From Microbe TV, this is Immune, episode number 19, recorded on April 16th, 2019. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello and welcome back.
It's the day after tax day.
It is. We always wait until the last minute.
Yeah, me too. I filed on the weekend.
That way they don't make money off of your taxes. So
unless you, because I haven't had a
because
refund
we always owe at the end.
Also joining us from Durham, North Carolina, Steph Langel.
Hi there. Happy April. We're midway in between. Are you past grant season? Are you traveling a lot? What does your April look like?
Grant season is continual every week.
It is continual. You're right. But Vincent, you had mentioned that March was particularly busy for you with 3 grants. That sounds like quite a bit.
Yeah, 3 grants rejected.
Oh no!
But they had been submitted, earlier. We got one in last Friday, and we have one every cycle, 1 or 2 every cycle, basically.
Right.
All the time.
Yeah.
There are a couple still incubating.
Hopefully they hatch positively.
I hope so. I'm still teaching my virology course, which is a lot of fun.
Oh, nice. And
I'm going to start doing a lot of traveling in a couple of weeks. As soon as the course is over, then I hit the road.
Great.
We have rock shows on the road. TWiV rock shows.
TWiV rock. I will have to look at our calendar and see if we're going to see each other for Immune like we did last year, but I don't know if we will this year.
You're not going to ASV probably. Right?
I'm not because I started in the new lab in January.
Yeah, sure.
I have data now, but the abstracts are due in February, so it's a little too early.
Right. All right. Before we start our paper, I want to remind everyone that we could use your financial support. If you like what we do, you could give $1 a month, which is not a lot. $1, one podcast for Immune anyway. But when you contribute, you help all the podcasts of Microbe TV. You can go over to microbe.tv/contribute. You can find out different ways you can help us. We'd really appreciate it.
And the money goes straight to the podcasts.
Goes to the podcast for our expenses.
Make it better.
And we travel. It helps us to travel. Right now, we're severely limited because travel is kind of expensive. But hopefully in the future, we can do more of that. We can come to your town.
Yes.
That'd be great. We need to put Immune on the road.
Yes.
Yeah, we'd love to do that. Yes. One day we will. One day the immunology meetings will say, hey, we should have that podcast here.
That's
well,
right.
I'm going to AAI this year, and I want to try and talk to people about it. And
where is it this year? Where is it located?
It is in San Diego.
Yes.
Nice.
Now, next year, it's in Hawaii. So
I know.
It would be maybe a little pricey for them to bring us out. But we should, I think, start reaching out and making those contacts and then build that, that relationship. And then maybe next year, a couple years down the road.
Mm-hmm. Yeah. So TWiV gets a lot of road shows.
Mm-hmm.
It's the oldest podcast. And then TWiM, we do one a year at ASM Microbe usually. But so we have to raise the visibility of Immune.
We do.
Yeah, sure.
Last year I took out ads, but our money is tight this year, so haven't done any yet. But
when I think having guests on too, I think we could start, each bring on a guest and I don't know, either at your university or we could have them Skype in.
All right, we have a paper today. It's my turn, and as you can probably guess, it involves viruses. I have to have a little comfort zone.
Right.
If I did something on, let's see, lymphocyte development, lymph node.
Somatic hypermutation.
And these two would be yelling. They would be yelling at me the entire time.
No, but you took an easier route because you picked a topic that's near and dear to Steph's heart. So
I know. Did you know that you picked something so close?
Yeah,
to
of course.
work?
I picked it because of you. Yeah.
That way he can get backed up there.
Yeah, there you go.
I knew you would like it for sure.
I know.
Plus, but we have also been talking a lot about herpes simplex virus on TWiV recently. Mm-hmm. We've been talking a lot about it in the context of Alzheimer's, but also we did a paper where they looked in neonates, and they tried to isolate herpes simplex viruses. And they had only 10 isolates, but they sequenced their genomes to say, are these any different from other circulating herpes? Is there something different about a herpes virus that infects a neonate? Which is a tough question because they spent 6 years and they got 10 samples.
Wow.
Right.
And was there a difference?
Well, there are, of course, but that's not surprising. Whether they mean anything is the tough part, right?
Sure.
So you could say, yeah, these changes look like they correlate with neurovirulence, but to really know, you'd need many more samples, but it doesn't look like that's going to be very easy to come by. So
No.
anyway, so we're aware of it, and this paper came out recently, and I said this would be very cool. It's in Science Translational Medicine. It's called Maternal Immunization Confers Protection Against Neonatal Herpes Simplex Mortality and Behavioral Morbidity. So I would just like to say it should be herpes simplex virus.
Yes.
Right.
Because herpes simplex is nothing on its own.
Doesn't cause mortality, those two words.
So, we have senior people on this paper. I know Don Cohen, David Knipe, David Lieb, all senior people, maybe others as well. I just don't know them. And they should know better. But people don't typically pay much attention to writing, which is too bad because it's the way you communicate, or an important way that you communicate.
Do you think that's a function of how quickly things have to get churned out?
I think so. But a title is like a key thing. You see, you look at it, you see it over and over and over at the top of your manuscript, so you would think. But they're probably used to saying herpes simplex, disease and no virus. So the authors are from a variety of places. Dartmouth School of Medicine, which is, of course, in New Hampshire. The Institute for Medical Immunology in the Free University of Brussels in Belgium, and then Harvard Medical School. And yes, that's all. And the first author is Shaya Patel, and then we have as the last author David Lieb. And David Knipe is a herpes virologist I know for many years because he also was a postdoc with David Baltimore.
Oh.
Now, did we do his paper about the chicken herpes virus? Marek's disease? Was that his?
I don't think it was
I don't
his.
think
no.
so.
Oh, okay.
No.
Okay.
It was the last paper I did, I think, right?
Yeah.
I have an affinity for herpes viruses here on Immune. So this is neonatal infections with herpes simplex viruses. There are two, herpes simplex type 1 and 2, and typically we say that type 1 gives you pharyngeal oral infections, type 2 genital, but that's not an absolute thing. They can go back and forth. But if a child is exposed to either of these two herpes simplex viruses, either during birth or as a young child, you can have serious infections. And about half of infected newborns, so some of these infections can be asymptomatic, but half of them can have disseminated disease and encephalitis, infection of the brain. And that's a problem because no matter what you do, even if you can control the infection, you're going to have long-term issues, neuro issues. And these kids have to be cared for, and it's a long-term economic burden, it's a family burden,
right,
and so forth. So this is a preventable thing. We have the skills to do this, and that's what this paper is about. So you can treat these children with acyclovir and antiviral, but even though you can control the infection, they still have neurodevelopmental issues. So this is a problem. And so the numbers, herpes simplex 2 causes over 70% of neonatal infections, herpes infections. But in the U.S., Europe, and the Western Pacific, type 1 is the major cause of neonatal herpes.
Do we know why? Is it just a geographical distribution and spread, or is there some other reason for that?
I don't know.
No?
I don't know if we know or not. No. The global incidence, 1 in 10,000 live births. So one neonatal herpes simplex virus infection per 10,000 live births. Although, it's regional, it can go up to 1 in 3,200 in some parts of the U.S.
Mm-hmm.
And so how do you get this? Well, so the mother can be, most people get infected young in their lives. often shortly after birth. And then you may have a primary disease, some kind of respiratory disease, and then the virus goes latent in your peripheral ganglia. And then you have recurrent, for herpes 1, you have recurrent reactivations where virus is made, and then you can have a fever sore on your lip or in many places on your head. And the same goes for type 2. And the interesting observation is that if the mother was infected long before pregnancy and has a latent infection and it's being reactivated, the chances of transmitting neonatal herpes are much lower than if the mother has had a recent new infection.
Right.
And the idea there is that's because in a latent infection, the mother's had the virus for years and she has antibodies that she can pass on to the child and protect the child from infection, maternal transfer of antibodies. And so this is the point of this paper. Can we take advantage of that and basically immunize the mom to protect the baby, which is something that's gaining traction in other pathogens. I talked with someone last summer who works on respiratory syncytial virus, a very serious pathogen of very young children. And hard to immunize them, but you can immunize the mom, and the antibodies will pass through the placenta and into the fetal circulation and protect the baby.
Yep. And there's multiple different pathogens that we think of that have had success with this maternal immunization strategy. So influenza is one where there's not a vaccine designed for pregnant women, but it is recommended that if you're pregnant, you can get the vaccine. Particularly, I think they see the most efficacy in the second trimester because when you do it matters. And tetanus has also been very successful at redoing tetanus infections in infants. And you're right, RSV, we're working with HIV, also rotavirus. So it's a good way, but you have to consider which antibody you're targeting. So what the correlate of protection is. Is it IgG that will transfer through the placenta, which we'll talk about here? Or is it IgA, which would not cross the placenta and maybe come in breast milk if you're thinking of an enteric pathogen. So it's definitely interesting. And RSV is tricky, I think, because of the way the way the antibody binds to the virus, at generating that specific immunity. But it's very exciting as a way to protect, because you're protecting both mom and baby in this way.
So for herpes, this hasn't been considered to immunize the mom. What has been tried is to make vaccines for preventing horizontal adult to adult transmission.
Now, I look back in the literature, and there is literature on this back in 2003, in the early 2000s, where they were looking at doing maternal immunization. So I'm not sure why it didn't result in a vaccine and is now popping back up.
Yeah. Well, maybe something didn't work, but
right.
It's coming back.
Right.
And in the meanwhile, there are a number of vaccine candidates for herpes simplex viruses that have been developed. So they took one of them in this paper, and they're going to be using that.
Why did they choose that one? So that one, is it just because it's a replication defective one, right?
So they have this virus it's an HSV2. It's called DL5-29, which has a deletion in two different genes involved in DNA synthesis. And it started with a mutation in one gene, and they felt that wouldn't be sufficiently stable, genetically stable. So they said, let's put a second mutation, and they do. And you have to grow these viruses that produce the gene products that are missing or that are mutated in the vaccine itself, right? Mm-hmm. So you can grow them up in these complementing cell lines, and then those viruses will infect cells, but they will not produce new virus progeny because they can't replicate their DNA. But it's apparently enough to get, an immune response. So that's the vaccine they're using here in a mouse model, and this previously been looked at in mice and guinea pigs. I think it's also in a phase 1 trial, in people. So HSV2, and also it's a big problem in Africa. So this is almost being targeted to Africa in a way, because the burden of disease in adults is huge of HSV2. So the first thing they did was asked if herpes antibodies to herpes simplex, IgG specifically, can be transferred from the mother to fetal circulation. And so they got matching serum samples from the mom and the cord, the umbilical cord, the 1-month-old children and 18-month-old infants. And they looked for herpes type 1 and 2 specific IgG. And they can find them in both the mom and in the cord and in the fetal circulation. In fact, the cord is pretty interesting because it seems to be concentrated there from the maternal circulation. They have a nice way of graphing this as a ratio, maternal to cord, and you can see what it should. If it were equally dispersed among the two, there's a line, and then you can go above or below the line. And they look at antibodies to a variety of viral glycoproteins, of which may be important for protection. So the mom has antibodies, the cord has antibodies, and you can also find it in the neonates. But at 18 months, they're gone, which is usual. Your mom's not going to stay with you forever. You're on your own. You're on your own.
Did they look only at babies that were breastfed or not breastfed, and did that make a difference? Because they do, they talk about maternal, transfer.
They didn't mention that. They didn't mention that
because that should make a difference, right?
Well, I mean, if they're only looking at serum, IgG in milk is low, so it would have to be suckled and then work its way through the gut and into circulation, which isn't as efficacious as the placental transfer. Okay. But I think though breastfeeding definitely impacts things, and that would be nice to see in this graph stratified out, if there's a difference. But the other thing is I would be interested to see which of these infants, and maybe it's just too hard to tell. I don't, diagnosing, but which of these infants got her HSV 1 or 2 infections? And can you then correlate the levels of transfer to whether or not they got it? And I'm just assuming it's because only 1 in 10,000 are getting HSV infections, or at least clinically diagnosed. So it's probably just not enough samples to match that. But that would be interesting because this kind of, we see this is very kind of just, I mean, I don't mean to, it is a neat graph, but it's like, yes, if it's in cord blood, it'll end up in the baby's blood as well. So correlating that to clinical disease would be interesting.
Yeah, but apparently this hadn't been done, so
be
done for herpes. But some of these antibodies are neutralizing. They actually do virus neutralization assays, which is do these antibodies block infection, and the more total antibodies you have, the better neutralizing they are. So they're high and low responders, and the high responders have, in terms of total antibody titer, they have more neutralizing antibodies. So the neutralizing antibodies, those are the ones you want, obviously, because they will block infection. They're transferred from the mother to the cord to the fetus. Okay, so that says there's nothing different about herpes antibodies,
right?
Which, is probably not surprising, but you had to do it anyway.
Yeah, it'd be interesting to know which one of those glycoprotein-specific antibodies was responsible for the neutralization. If they were to stratify that'd be neat too.
Yes.
So I looked in the methods, and they looked at 24 mother newborn pairs, and then they were able to collect at 1 month from 14 of the kids, and at 18 months from 10 kids. So I would think they would have been extremely lucky to get an HSV infection in that few number of individuals. So it would have to be a much larger study.
I agree.
Yeah, it's hard.
Sure. Yeah.
All right. So now we move on to mice, and we answer a couple of questions. First of all, can antibodies get into the peripheral ganglia? So in the herpes simplex 1, which you get as a nasopharyngeal infection early in life, it typically becomes latent in the trigeminal ganglia, which are two ganglia on either side of your head, which are collections of nerve bodies. They're outside the central nervous, so they're peripheral nervous system, and they innervate your face, essentially. So the virus genome goes into them, and it remains quiescent, quiet, And then when you get stressed, I tell my class, when I give you an exam, you get stressed and you get herpes reactivating in your trigeminal ganglia. And because that ganglia innervates your nose, your face, your lips, you can get cold sores all over. Or some people get reactivation without any cold sores. They shed virus, and then, of course, they spread it readily because they don't know they're shedding it.
Right.
So here in this study, they want to know in mice where we can establish latent infections. You can infect mice with human herpes viruses. There are a number of ways to do this. One way is to simply scarify the lip and infect with the virus. It will then go to the trigeminal ganglia and establish latency. And latency, again, means simply there's no virus being produced. The DNA genome is in the ganglia and it's quiet. So they latently infect their mice. So you infect the mice and you wait a couple of weeks so there's no virus produced. And, you could look in the trigeminal ganglia for virus, but it's there. And then they ask, can we detect antibodies there? So first of all, antibodies in the sera. There's clearly, if you have these latently infected mice, you can find antibodies to the viral glycoproteins and also to a non-glycoprotein of the virus as well. And you can make extracts of the trigeminal ganglia from these mice. And you also find antibodies in those as well.
So I was kind of surprised by this, and I don't maybe don't know as much about antibodies as I thought I knew, but I thought it's extremely difficult to get antibodies into the brain.
I'm glad you said that, Cindy.
There's therapeutic antibodies that people have been trying to deliver into the brain, for example, against Alzheimer's, to bind up the proteins that are congealed there and things. And it's extremely difficult to get them in. And people are studying how to break the blood-brain barrier or the blood-CSF barrier to get these things into the brain. So I'm actually shocked these antibodies can be found there.
Well, this is not the brain, remember. This is a peripheral ganglion.
Yeah,
So maybe there's similar issues, but I think it depends on the antibody, because I remember Diane Griffin years ago reporting they could treat mice with an antiviral antibody and it would get into the central nervous system and prevent neurological disease, but it was a special antibody. So maybe that's part of it. But here they're in. I was surprised also, but they seem to treat it as, we looked and we're there.
Yeah. And maybe similarly, I was like, oh, wow, there's antibodies there. But I don't think enough about the brain and nerve immunology. I'm mostly in the gut and the mammary gland. So I'm thinking, I just need to be thinking about the brain more, because that's fascinating. If we can treat, herpes virus infections with, I'm sure they've tried this, but antibody infusions that can end up in the trigeminal ganglia. It's interesting.
So I don't know how important that is, because
it would be hard to determine, because don't they say most of them are asymptomatic and then they go latent, so you'd have to catch it. You'd have to know. And
also, as the virus is replicating in the trigeminal ganglia, it then leaves,
right?
It goes out the nerve ending, right? And it goes into the mucosal epithelia. So if you have antibodies outside of the PNS, it's going to be fine to neutralize virus. And there will always be viral genomes in the trigeminal ganglia. Right. You're never going to get rid of those, no matter how much antibody you get in there. It's never going to get rid of the genome. So I'm not sure how useful that is, but they looked and they find it there, and that antibody is neutralizing. It can block infectivity.
Right. But I guess what they're suggesting is, at least in this case where you have neonatal contraction of this disease, if you can prevent it, then you're fine. If the antibody is already there, that's okay. Gotcha.
But they look both in sera and in the trigeminal ganglia, and we don't know which. If you took away the trigeminal antibodies, I bet the outcome would be similar. Right, but we can't do that experiment. So they also use mice lacking B cells, the MUM-T mice, which we've talked about before, and those animals, of course, do not make antibodies to the virus, so they can infect those with herpes simplex virus. They get latent, but they don't make antibodies. So this is obviously a B cell specific phenomenon. So again, the point here is that they're latently infected and they have antibodies both in the serum and trigeminal tissues. All right. So what about mother and child. So what they do here is they first they try do serum transfer experiments. So if you had a latently infected pregnant mouse, and you gave them serum that's known to have antibodies to herpes simplex, would that prevent disease in the offspring if you challenge them with herpes simplex virus? Okay, so they have pregnant mu-MT mice, which have never seen herpes virus, and they give them serum, either immune anti-HSV serum or control sera before birth. And then the pups are challenged with herpes 1, simplex 1 intranasally. So if you do this experiment with pups from mothers that got immune serum, they have lower viral titers and they're protected from death. And if you do it with pups, if you challenge pups from mothers who got the control serum, they get widespread virus titers, and that's in the brain, the trigeminal ganglia, visceral organs, and they die. And they can give these mothers purified IgG, and that gives you the same effect. So you can give the mother antibodies, and those antibodies will protect the pups from a challenge of herpes simplex virus.
Right.
So the implication, right, is that they're being transferred from mother to pup, and that's protecting the pups.
And when you said something earlier, Steph, you said in humans, when they're talking about immunizing moms, for example, it matters when you do it. So do we know, as far as in a mouse model versus a human, what the comparison times are? And I didn't look exactly when they were injecting these dams with the immune sera or the IgG, but does it make sense as to when it would be transferred?
Right. So what's interesting is it says that, they administered them prior to parturition. I'd have to go back and look. I feel like it was 3 days prior, because the IgG will decrease quite rapidly. And the latter part of pregnancy is when you have the best transfer transplacentally. Now in humans, they find that seroconversion by vaccine is best in the second trimester because it gives the body a chance to ramp up that response and then transfer things in cord blood. In mice, it's 21-day gestation, and you can break that down into 3 different sections, but it's not it's not as well investigated whether the second trimester would be best for a vaccine, because really in that case we're talking about B cell development. For this, you just want to infuse it in the latter part when the placenta is most readily able to transfer. For a passive transfer infusion, just as close to parturition as you can get, but for a vaccine, you would need to do it earlier.
So giving antibodies to pregnant mice protects the offspring. What about immunizing now? That's the real question. Right. So that's the next experiment. Can maternal immunization protect the newborn mice? So they use this vaccine, which we mentioned, herpes simplex type 2 now, and it's DL5-29. It's a virus that doesn't replicate because it lacks genes needed for DNA replication. It has been studied in animal models. It's been studied in phase 1 in people. And all that was done with subcutaneous immunization, which they say is not as good as intramuscular. So we're going to do IM immunization in our work here. And so the previous failures of this vaccine, they say, may be related to the route of immunization. So they're going to take pregnant mice and immunize them intramuscularly, and you either give them virus or a control which doesn't have virus in it, and you're going to see if they make antibodies and if those protect their offspring. So they do the experiment, they immunize the pregnant mice, and they look for IgG antibodies in the circulation. So they find that in the mothers, you find antibodies against the viral proteins as before, and serum from the offspring have antibodies against the viral proteins as well. And those are all higher compared to the experiment where we don't use virus.
So one of the things that was interesting looking at this was, the viral titers in the lung are quite high, and they mentioned that that's because it's an intranasal infection. But one of the things that would be neat to see is if I could see viral titers on a time course scale like they do for the survival, because I want to know if those lung titers are able to resolve themselves or if they become latent, if that spreads to other parts of the body, because maternal IgG could prevent death, which they show, and it could decrease titers, but is it sterilizing immunity? So, I guess it wouldn't be immunity because it's not a vaccine for the pups, but does it totally prevent latent infections? Be interesting.
Yeah.
Because we don't know 20 days. Yeah. Are there still titers there? Was it effective at completely removing any viral titers? Because that's our problem, right? Is not necessarily, the survival is important, but you do not want any viral titers to reach the trigeminal ganglia. And we don't know that.
Yeah. I don't know. I think if you wanted to prevent latency, you'd have to knock it way, way down. I don't know what the lower limit is, though.
Right.
That's a good question. Yeah.
Yeah. So I think this is great because it also shows they show HSV 1 and 2 to try to compare. And I was reading, and actually Gertrude Ray, who writes a lot for your virus blog, Vincent, she wrote about HSV vaccine candidates and said that the mouse actually isn't a great model for HSV because it does not spontaneously reactivate. So the latent model isn't great. So I think this is interesting to prevent possibly death, but in terms of, looking at, we may want a guinea pig, which she said is a bit better.
Yeah. Yeah, you have to heat shock the mice to get reactivation. You put them in a water bath. You put them in a warm water bath for like a half hour. And then we just did this paper on TWiV. You have to then keep them under a heat lamp for a half hour so they don't get chilly.
Huh.
So and then that reactivates the virus.
It does. Okay. Interesting.
Yeah.
Do you think that's partly because the mice are always, remember the very first paper we did, we were talking about heat stress and cold stress.
Yes.
Right?
Yes.
And the mice are kept much warmer than they normally, or much colder than they normally should be because of the being comfortable for people. So they're kind of cold stressed all the time.
Huh.
I wonder what would happen if they weren't cold stressed all the time.
Also they're nocturnal, but we don't treat them
That's true.
that way.
That's true too.
Right.
Yeah.
We put the lights on in the day in the mouse facility so we can work them.
Right.
Right.
But I think they must be all screwed up.
So
And
they're
the
all
thing
stressed.
is when you look at a mouse, its facial expression never changes. It's always got that same deadpan look on it, no matter what.
Right.
So it's kind of hiding what's going on inside.
Yeah.
In this experiment, so you immunize the mother has antibodies to the virus in the serum, so do the pups. They also look in the trigeminal ganglia of the pups, and they actually stain for IgG in the trigeminal ganglia, and you could see them there. So it's transferred to the pup, and it not only in the pup serum, but gets into the trigeminal ganglia. Okay, so now let's try a challenge experiment. We immunized the moms, and then we challenged the pups. And they challenged them intranasally with lethal doses. They show that the pups born to immunized mothers have lower viral burden in the brain, the central nervous system, as well as the peripheral nervous system and various organs. And they survive the challenge. And the pups born to mock immunized mothers die. They also do this with as Steph mentioned, a type 2 isolate, clinical isolate of herpes type 2, and they get comparable protection, which goes for 4 pregnancies, which is over 245 days. So you just get one immunization, and it lasts for 4 pregnancies.
Yeah, that was impressive.
Pretty good,
That's
right?
pretty good.
I thought that was amazing. To me, that would suggest that there's always a little bit of reactivation at a low level so that you keep triggering the production of the antibodies, because I would think that the titers would drop over time, especially if the virus is latent and there's nothing being produced.
But they don't reactivate, so that's
you don't need a lot of antibody, right? You have pretty low titers, once that initial high level is there. And we know from epidemiological experiments that you can have no virus around, those measles experiments in the islands north of the UK, what's the name of those islands? The famous one.
Aleutian?
Oh my gosh, I can't believe.
Aleutian? No.
No. My gosh, I just lectured about this. I'm forgetting everything. Anyway, there's
your students can tell you.
They're up there, and nobody goes there because there's no airplanes. It's a long time ago. They have an outbreak of measles brought in by shipping, and then nobody comes to the island, and there's no measles for the next 60 years, and then someone brings measles in, and everyone who had measles the first time is immune the second time, and there's been no virus in the interregnum. So you probably don't need to have constant challenge with virus.
Shetland
Maintain a low level
Islands?
of not Shetland.
Or
All right, I'm gonna go right now and look at my lecture notes because it was only yesterday that I gave this lecture, and I am failing miserably.
So how do
my
you,
mind is going.
that example, is that because the population of the island was so low and they're not densely populated? Because our measles outbreaks here would suggest that a break-in vaccine, 60 years, they would not be protected. If measles is brought back.
Oh, because there's no influx. There's nobody bringing measles into the population. It's quite small, yes, so that you don't have measles circulating all the time.
Okay.
Yeah, here we have so many people that the virus is always finding hosts, right?
Right.
But it's not the case there. All right, let's see.
Islands?
Faroe Islands, thank you. Very good. Faroe Islands.
Don't
famous
thank me,
experiment.
Google Maps.
Thank you, Faroe. Yeah. I can't remember the simplest things. Oh, well. So I don't think they need to have reactivation. Anyway, then they want to know if these antibodies are coming through the placenta or from, milk and through the mammary. So they take the babies when they're born and they give them to a foster mother who doesn't have antibodies to herpes. And they find the protection just as good. So they say that's the placental antibody that's doing this. So how does that sound, Steph?
Yeah, no, that would be good. It would be dependent on IgG transfer, not IgA, which can't transfer through the placenta and is majorly in milk. Mm-hmm. So this would be, IgG seems to be the correlate of protection for the pups.
So if it were IgA, when they fostered these pups, then they would get
better protection.
Less because they're being fostered on a non...
Oh, I'm sorry. Yes. Flip it. Yep. They would be... Yeah. Yes, correct.
Okay. And so far, they haven't even measured IgA, right? So
no, they didn't.
Looking at IgG. So.
Yep.
Anyway, so you can protect if you immunize the mother. Now, the last experiment is quite interesting. Then they say, let's look at the behavior. Now, as I mentioned earlier, infants who get neuro-HSV can have long-time neurological issues. So they wanted to make a mouse model to study this, and there is a test called the open field test. It's a behavioral assay, which you can use in mice, which basically measures where they are in the cage and how they explore things and their reflections of general anxiety levels in the mice. So, for example, if you put mice into a new environment, they will typically explore. Unless they're stressed, then they will stay close to the cage walls. This is called thigmotaxis. I wanted to ask you, Steph, do pigs exhibit thigmotaxis?
That's a great question. No, not that I have observed in my time working with pigs. But they are not, they're not a prey species. So I'm assuming that, but then they say they observe it in humans as well, in terms of anxiety levels. So I'll have to dig into the swine behavior literature for you to get back.
It's interesting. I view pigs as being really self-confident. Nothing's going to stress them out.
Yeah, they can get stressed, but they're very self-confident.
And they're not a prey species in the wild, you say, right?
Well, they can get preyed upon by, I guess, larger dogs, coyotes, wolves, but they're not like the way I think of mice.
Yeah. So a mouse, when they're stressed, they stay by the cage wall. So they ask if herpes infections has any effect on this open field test behavior. So they take the offspring of latently infected or mock-infected mothers. They challenge them with a very low dose of herpes 1 so they don't die. Right? So there's just a low dose, and the mice will remain living. And they look at their behavior till about 5 weeks. And offspring of of the mothers without immunity, they had thigmotaxis. They displayed thigmotaxis. They stayed near the periphery of the cage. So they're stressed.
Yep.
Presumably because this virus is in this nervous system, and it's wreaking havoc there. And now you take the offspring of latently infected mice. They were fine. They explored both the walls and the inner parts of the cage. They walk all over the place. And so and we know already that they're protected from infection. And then they ask whether immunization of the mothers could prevent this kind of behavior. And so you mock immunize the mothers. They have pups. You put them in the cage. They are also stressed. And if you immunize the mothers with the vaccine, they have normal exploration of the cage. And so the interesting thing here is that even at a low dose of virus, a far sublethal dose, you get behavioral aberration, and that can be prevented by immunization.
Yeah, this part was the most, I found it fascinating. I often think about, the consequences on behavior. And so if we think about antibodies and viruses that potentially in a human context affecting the behavior of children and their development, it's definitely sings praises to maternal immunization if we can prevent things like this from happening.
For sure.
Yeah.
I mean, I think a lot of behavioral abnormalities, cognitive issues, could be related to low-level herpes simplex infections that we're simply not aware of.
Right.
We're aware of the serious ones that give rise to encephalitis because we can tell they have symptoms, but maybe there are low-level things and low-level infections that cause issues. And if we immunize the moms, we can protect them all.
Well, it'd be interesting. I know that there's some literature suggesting inflammation during pregnancy can affect fetal development in the brain and autism.
Yeah.
So, Vince, I remember you discussing on TWiV about treating people with, is it acyclovir, high levels for Alzheimer's?
Mm-hmm.
You wouldn't really, I don't think you would want to do that in pregnant women, but who knows? I mean, it would be interesting to see.
Yeah. I think a long time ago on TWiV, we talked about a paper, a study done in mice where they used influenza virus and showed in pregnant mice this causes issues in the offspring. And the idea is that, inflammation triggered during pregnancy is bad for the fetus.
Right. So
fetal development.
Vaccination is even more important, especially during pregnancy, if that is the case. We have to.
Absolutely. I mean, they say here that timely acquisition of immunity to a pathogen can lead to considerable benefits for the offspring. The maternal virome is therefore likely an important determinant of human health.
Yeah, I liked that statement because you hear a lot of arguments about, the natural infection is better, you have better immunity. But this is one of those times where if they have the natural infection, this is one of those viruses that can go across the placenta and infect the fetus too, right? So if you had the infection with the wild type virus compared to a vaccine that will then drive protection, at least during pregnancy, that's a big deal. So if you have a mom who hasn't had the infection and doesn't have latent, doesn't have latent virus and doesn't have protective antibodies, this would be a good reason why they really should have an immunization to protect the baby.
I think by the time you're having a baby, you're likely already to have herpes. So I'm surprised that there are women who have recent herpes while they're pregnant. Right? That's unusual. But
I mean, you could have HSV1 and then acquire HSV2.
I suppose. Yeah. But the point is there that you'd want to have either had it. And the thing is, you don't know, right? You don't know if the mom has been infected or not. So you just should immunize everyone.
Right. And the other thing, we don't know the transfer efficiency of these antibodies, and there are viral infections. So HIV can actually cause placental transfer to decrease because of abnormal activation of the nonspecific B cells. And so if you have co-infections, you may not transfer HSV antibodies as well. And so this paper, I really like it because it kind of gives us more information. Now, one part that they could include in here is, does this vaccine prevent infections in the mother? Because if it only transfers the antibody but doesn't prevent an infection, a primary infection in the mother, then you could increase the transfer of virus. So that would be a nice other side of the story, I felt, for this paper.
Yeah, that wasn't their focus.
Sure.
Right? But the infection with herpes as an adult is far less serious than in neonates.
Right.
So there's less of urgency to protect the mother from infection, which is not to say that you can't get encephalitis, but it's much rarer than if you're infected as a neonate.
I guess I'm just speaking to that 55% is maternal primary, so latent wouldn't be. But anywho, but it is a good paper, and I'm glad you picked it.
They also mentioned at the end that besides vaccines, you could do passive antibody therapy, right? Which they say is being explored because we're getting really good at making therapeutic antibodies now that last months in the serum and which neutralize very effectively. So I guess what would you do? You have a pregnant woman, you could give them the serum that would then get transferred and protect the neonate without vaccination, I suppose. And that could get developed before a vaccine as well. So that would be really interesting. One other thing I wanted to say, what was it? You were just talking about protecting the mother. Oh, so what if this turned out to be safe and effective? When would you vaccinate? When you got pregnant, you think, or before?
So I think it would depend on the robustness of the antibody response. But in terms of what we know from influenza, its second trimester vaccination seems to be an ideal time to at least boost the immune system to get peak titers by the time that they're able to cross the placenta at the maximum rate. But I think you may be considering HSV. I mean, you would probably want multiple boosters. Maybe before you're pregnant, one during pregnancy.
Well, I look at the figure where they have the pups that were challenged from the latently infected or the mock infected moms. I mean, that's after they've established the infection and developed protective antibodies. So in that case, I would think, and the immunization too that they did, that if you do it far enough in advance, there's going to be antibodies around because these antibodies stuck around for several pregnancies, right? So as long as you establish that level of antibody, it should be lasting. So there shouldn't be any requirement to wait for pregnancy, I would think.
I know. And that's interesting in terms of why it doesn't work that way for influenza. So why do we have to boost? Why does it? Well, I guess it would be the virus, right? Because we get different strains every year and there's only 2 serotypes here. So that could be a virus specific thing.
And you have an RNA virus versus a DNA virus and you have latency versus non-latency. So that was one of the things I was going to ask is there anything that we know of that would inhibit infection completely? So prevent establishment of latency for a DNA virus that works latently.
I mean, you could stop. See, even if you stop it in the neonate, they're eventually.
I think they're still going to get. I think that even in these mice, the neonates are going to end up with a latent infection.
Yeah, right. Just based on the titers, especially in the lung, I don't think that. I mean, I don't think that would just go away.
Because I wonder.
And maybe that's.
Yeah. Go ahead.
Well, I was going to say maybe that's why they're having issues with a vaccine because they can't get sterilizing immunity. And it's always going to become latent.
Yeah, so I think I read that too, because one of the things that they look at for success of a vaccine is preventing infection, right? And so in this case, you can't prevent infection, but what they're arguing is that you could prevent the sequelae that happen because of the infection. So
like
that
the
this
severity
is still
of it in a susceptible population.
So this is still a worthwhile vaccine, even though you can't prevent infection. But we have one of these already, right? Because we immunize against chickenpox.
Right.
And
and that does prevent latency.
It does prevent. So that's why I was going to ask. It definitely does prevent latency.
It's like 99%. Yeah, very rare to get shingles in a vaccine related shingles. I was just looking it up today because yesterday someone asked me in class if you can get shingles after the vaccine. So I looked it up and it's extremely rare. 99%. So maybe 1% of the people who get the chickenpox vaccine will get shingles.
Now, that's the childhood vaccine.
Yes.
And so this has been long enough, and we've had long enough people who've gotten the vaccine to know whether or not they're reactivating.
So that is what I told the student. I said I didn't think it was long enough.
Right.
But I looked it up, and they quote a infectious disease person who said it's so far it's there's very little shingles, although maybe it will change as we go further down.
Yeah, we'll have to find that out, because I know when
what's the name of the vaccine? Do you remember?
Heard about it.
Childhood one, I can't remember.
Varicella vaccine. Also, let's see.
Zostavax?
What is it called? Because then I can see Zostavax. That's the zoster, isn't it?
Yeah, I think you're right.
Let's see when it was introduced.
The 90s?
Developed in Japan in 1974.
But when was it broadly?
And it was licensed in the U.S. in 95.
Right. Because I remember I did.
How many years is that? So that's 19, let's say 25 years.
So really, we have to wait
time.
like 50 more years to really find out.
It's not long enough.
Right.
Yeah. Because those kids presumably got it when they were, 10 years old or something. I don't know what age you give the chicken pox, so they're still not old. They're not over 50 yet.
No,
I mean, that would be because I didn't get the chicken pox. I think the vaccine was really starting to be implemented right after me. And so,
And you're not 50.
30. I'm 30. Have to wait a little bit.
Wait a while.
I did not get the vaccine. I had chicken pox as a kid.
Yep.
And then I had zoster. The first year I started TWiV, I got zoster.
Oh, that stressed you
I
out.
talked about it on an early episode.
I know someone who just got it recently. This is sort of a tragic thing, and the reason why we really need people to be immunized is so this individual got really stressed and had been waiting actually for getting the new Shingrix vaccine, because he just turned old enough to get it, and it was back ordered and nobody has it around in its stock because it's so popular, right? And so he ended up coming down with shingles, and his wife just a few years ago had battled breast cancer and ended up with a primary case of chickenpox because her immunity was gone.
Oh my gosh.
So she's fine from breast cancer and now fine from this, but it knocked her down because a primary case of chickenpox as an adult is pretty bad, but she just had no immunity left. And so that's kind of scary and is exactly what we're always saying, like cancer patients or people with immune deficiencies. It's like we need to be immunized to protect them. And there's a very good example.
Were your kids vaccinated?
Mine were
for chickenpox. For chickenpox, okay.
Yeah. But I'm reading in the news now. I mean, this thing, like I thought it was a joke a few years ago with these chickenpox parties, but people are really honestly doing this, and this is insane.
Well, I mean, it's really a sign of privilege, because if you can be okay for your kid to be out sick and you don't have to, take off work, that's like good for you. But
The governor of Kentucky said he exposed all his kids to chickenpox.
Yep, exactly. That's what I read.
He said, "Oh yeah, they got sick, but they were okay."
Yeah, because he doesn't take care of them.
And also he's lucky that they didn't die. Right?
Yeah, very lucky.
I mean, that's ridiculous. That's why we made vaccines, to avoid the potential lethal consequences of viral infections. And I don't understand why people don't get this. And people say, I should have a right what to put in my body. But no, if you're going to endanger others, as the story Cindy just said, you don't have the right not to take the vaccine. If you want to not be vaccinated, you have to stay home all the time.
And that's now happened too, right? So in Rockland County and
here in New York City,
they've made a ban. They have if you're not immunized for measles, you're not allowed in public spaces right now.
Yeah. Unfortunately, that's been challenged by another court. So I don't know what's going to happen with that. But in Brooklyn, the city said, you cannot not get the vaccine. You have to get it.
Wow.
And if you can't prove that you've gotten it, you have to be vaccinated. So I was talking about this yesterday in class. I don't know how they're going to enforce that, but maybe just the edict will get enough people to be vaccinated,
right.
Because, in Brooklyn, there's a rabbi who's a spiritual leader, and he apparently mistrusts MMR vaccine. And so everyone in his community follows him. And I was telling the class, why does he mistrust the vaccine? What science does he know? He doesn't know anything. He's heard some rumors, and now he influences all these people. It's inappropriate. It's completely inappropriate. And, we happen to know this happens often in these religious communities. They have a leader they follow, and this is what you have.
Well, and based on what we don't know about herpes viruses, I mean, we don't know if it can affect down the road, as we discussed, Alzheimer's, infecting your own child. So, I mean, yeah, if I can get a vaccine to prevent herpes virus infections, I'm getting it.
Yes, you just don't know, right? And even chickenpox, we just don't know what the long-term consequences are. So why not just avoid it and getting vaccinated? Measles, we know, can go in your brain. Mumps, in half of kids who are infected, the virus is in your brain. And we always say, doesn't seem to make any difference. Well, maybe it does, and we haven't figured it out. And of course, now that we immunize, it's harder to do. But why take the chance? I mean, unfortunately, we're preaching to our audience here is converted. The people who need to hear this are not listening, and that is a real problem. That is a real problem. It's just it's a difference in the way we're scientists. The way we think about things and risk and so forth. And I wish we could just convey it to more people.
Well, our listeners can be our ambassadors and go out.
Well, that's what I told my class yesterday. Each of you go out and tell 10 people all about the greatness of vaccines. That's one way.
I think you have to prep them, though. We can't just give them these science facts. We have to give them. Because if you come up preachy,
people do not like that. Right. So they want to feel like you actually are engaged in kind of their well-being, and that unfortunately just takes time, building trust, and
it does. Yeah,
it's not a quick thing.
I think we should throw them all in jail.
I know you do. I was gonna say.
No, I just
different.
I like to preach, and I know it doesn't work, but. I'm going to preach. They preach bad stuff. I'm going to preach good stuff. The religious leaders preach the vaccine is bad. So why can't I preach that it's good? And they listen to the bad preaching. I don't get it. I understand what you're saying, Steph. You don't.
I know.
But
no, I know.
I think you need to have some passionate people making noise as well. Mm-hmm. I also think we could use celebrities because they seem to be listened to more than us,
which is a little scary.
It's very scary. Very scary. Anyway, that was a very nice paper. Hope you enjoyed it.
Yeah, it's good.
I liked it.
Me too.
Let's do a couple of emails here before we say goodbye. Cindy, do you want to take that first one?
Sure. So Bob writes, in Immune 14, the team investigated the mechanisms behind activating T cells to attack tumors. They discuss a discouraging fact that each potentiation seems only to work in about 20% of clinical cases. So this was the one where we were talking about the breaks, the PD1, PDL1, and immunotherapies in cancer. And it says there seems to be new work on a mechanism which could help alleviate this problem. And so he points us to a paper that shows that a protein called ADAR1, which is A to I editing of interferon-inducible RNA species, this protein is reduced, leading to double-strand RNA ligand sensing by PKR and MDA5. So these are mechanisms to sense double-stranded RNA. And what happens is this results in growth inhibition and tumor inflammation. And so those are known nucleic acid sensors that are important in viral recognition. And so they induce the inflammation and produce interferon. And so somehow loss of that protein overcomes the tumor's resistance to the checkpoints. So that's like a lot of mental gymnastics to do there. But tumors normally are resisting the checkpoint blockade. And this paper is showing us this loss of this RNA editing enzyme overcomes this resistance. So the idea here is like, could you possibly take advantage of this knowledge to understand how all this works? And Bob writes, with my limited knowledge of immunology, I'm unable to understand how this works, which I'm not surprised because it's really complicated. He asked maybe the panel could take up the paper announcing the research. So I actually think this is a really interesting thing, and it brings together a couple of different things we've talked about. And so I would put this on our list of things to talk about because it talks about nucleic acid sensors and inflammation and cancer and checkpoint resistance and RNA editing enzymes, which have to be interesting to virologists, right?
Yes.
So the way I understand this, so ADAR is our cells make some double-stranded RNA.
Yep. Mm-hmm.
And ADAR, modifies it chemically so it will not turn on, will not trigger innate responses, right?
Right.
So apparently having no ADAR leads to inflammation as a consequence of our own double-stranded RNA being sensed, and that helps get rid of
the tumor,
right? Mm-hmm. Yeah. Okay. So
I guess if you could target that enzyme, then the tumors would sort of recognize their own double-stranded RNA.
So you could do it locally, maybe in the tumor, right?
Mm-hmm. Mm-hmm.
Maybe so the CAR T-cell therapy, you're delivering the proteins via a lentiviral vector, right?
Mm-hmm. You could just encode that in.
You could put it in there, and then when you fuse it back in, maybe that would do it locally in the tumor. That's quite interesting.
I don't know what would happen if you add an RNAi to ADAR to see if you could block it. Whether that would also then trigger the response through the RNA recognition pathway.
Right. I guess, Cindy, I think you're next week. You could do that if you wanted to.
Next month, yeah. Mm-hmm.
Next month, sorry. Yeah, not next week. We could do it next week.
That's scary.
Sorry.
Wait. I know the listeners would love to hear it every week, but
I know.
It's kind of hard.
No, I don't know if we're ready yet.
Not quite yet.
Steph, you want to take the next one?
Sure. Sarah writes, "Hi all, just wanted to write in and express my appreciation for Steph's pick, The Pregnant Scholar, in last episode. So I think this is 2 episodes ago. I checked it out and it looks great. Great initiative. Keep up the great picks and episodes. Cheers, Sarah from Christchurch, New Zealand." And yes, I had picked, that was The Pregnant Scholar and its initiative to work with universities and individuals who are potentially getting discriminated against based on their pregnancy status or, parent status. And I've been keeping I keep a little document of stories that my mentors, other women in science, have told me of things that they've been told while they're pregnant or while they were a mother. I don't know who raised these people saying these things to women, but it is, really shocking, some of the things that older women in science have had to deal with. So I am just hoping that I can, in whatever way, help the next generation of female scientists. Not have to deal. People are going to say things, right? But that there's some type of mechanism that people can get help, I think, is the way to go.
You should keep collecting those stories.
Oh, yeah.
Does that
Cindy, if you have
think about writing a book about it or blogging about it.
Yes, I think that would be a good way to go. And I don't know if it would be, I think it'd be interesting. I think people don't know that maybe some of the things that are said, but a lot of it circulates around, if you get pregnant, you won't have, science, you won't have a career in science. Like you won't be able to work hard enough. But like, Cindy, you're a perfect example. My current boss, my past boss. So it's definitely possible. Cindy's rocking it out. My boss is rocking it out. You can definitely have kids and have a career in science.
Yeah, my wife had 3 kids and
yes, she had a great career.
She'd do it over again too.
Yeah. Right. It's hard, but it's rewarding on every level. So it's worth it.
Yeah. Well, hard is not, I always tell people nothing good is easy.
True.
It can be, but if you have to work hard, that's not a bad thing.
Right.
And sometimes it is more fulfilling. But everyone has their own thing. Anyway, we shouldn't preach to each other. You have to keep reminding me that. One more from Garrett. Thanks for all you guys do. It's nice to finally have an immunology podcast out there. Makes the commute more educational. I'm an MD PhD student in Texas, and I just wanted to maybe offer a slightly different perspective on some of what you were talking about in episode 14, Breaking Bad. You've been talking about immune checkpoint inhibitors and cost and things, and one of your comments was about the U.S. government not recouping some of the money from research. And it is a valid question, but there's another side to it, I think, that doesn't get talked about as much. One of the largest things that the U.S. government does is try and maintain a healthy, productive economy that keeps everyone employed, keeps the wealth moving and growing in this country, and frankly, every country. When the U.S. government invests money into scientific research, they are doing exactly that. That grant money pays for researchers and their staff, but has also created the entire cohort of scientific companies who produce reagents and components for scientific research. Those would not exist without the NIH grant money. These companies, in turn, employ many people, pushing money into the economy. When a therapy is developed that is marketable, researchers turn that into a patent and sell it. This research almost exclusively occurs at the levels of universities, and selling the patents to private companies brings valuable funding to the university to help fund its programs, provide education, and invest in additional research. And a pharmaceutical company itself is going to employ a lot of people as well. If the U.S. government were to recall that grant money, there's a very real chance it would dampen this impact on the economy, causing more harm than good. By just letting that money go, they provide the funding that fuels entire industries and at the same time advance healthcare and medicine and knowledge in general. Not trying to defend a pharmaceutical company here. They've got enough money to lobby for their own defense, just trying to put out a slightly different perspective than is usually talked about. Keep up the good work. So my view on this was, in that episode, was influenced by Mariana Mazzucato, who's an economist in England. She was interviewed on the podcast Freakonomics 348, and she was talking about how the government drives development but never gets any payback on its investments. And she cited NIH as one of the examples, but she had a lot of other examples as well. She said, NIH has funded the vast majority of all new drugs approved by the FDA, but gets nothing in return. And her point was, because the drugs are really expensive, the taxpayer pays twice. They pay for the research, and then they pay for the drugs.
That's right.
That's totally unfair. So her position is some of the profits need to come back to the government, which can then reinvest those. We're not taking back the grants, as you said here, if you take back the grants. That's not the point of it. Mm-mm. You want to get back, any company that invests gets something back, right? You never just invest and say, oh, keep your money, it's fine.
That's right.
The government does that. They say, oh, and they don't just fund biomedical research. They funded the development of the Tesla. Mm-hmm. They funded the technology of fracking and lots of other things, and they let all these people keep it and not do anything. So that was at least my perspective there. Having listened to her on it, was all her idea. And in fact, I'll put a link in the show notes. There's a transcript of the episode so you can read about what she's saying. And in fact, she gets a lot of criticism for this, but I like her position. I think NIH should get something back and reinvest it, not to just keep it, but reinvest it, right?
I wonder who makes up the people that are critiquing her. It'd be interesting to know their backgrounds and interests.
Yeah, I don't know. I don't know. Her interviewer said you get a lot of pushback, and she responded to that, but I don't.
Yeah.
I don't.
Just I have theories.
But I don't know. Her position's compelling, and I don't think it's going to do any of the bad things that Garrett is saying here. That's not what we're trying to do. We don't want to inhibit that. We want to make it even more. Get the government some more money so they can reinvest more. Because right now the NIH budget is paltry.
Yeah.
And for the amount of things that we could do, $35 billion is nothing.
I know people are surprised by that, but
I mean, it's a lot of money, but
it is. But considering the defense budget,
and considering what NIH does, and we could do much, much more, and a lot of good talent is being wasted. It really and so the federal government, its budget, our budget, can't accommodate more. Okay, fine, then get back some of the money. And a lot of these pharmaceutical companies make a ton of money. They pay huge salaries to their CEOs, which is just frankly not needed. They should put it into research.
Yeah, well, that's what I was thinking. The people who are actually doing the work, right? So they get trained on NIH grants, for example. They get their PhDs. They go out into the workforce, work for a pharmaceutical company. They're not the ones making the big bucks, right? It's the business executives.
Right, for sure.
So they charge these exorbitant amount of money for these drugs that they develop, but then it doesn't really trickle down to any of the people who are involved in the process anywhere along the way. So yes, it creates jobs, but it really pushes the money up to the top.
Anyway, Garrett, that's what I thought. And you should listen or read her interview and see what you think. It's not limited to NIH and pharma. It's all the investment that the government does. Yeah. They just don't.
And I don't think he's wrong for bringing up this point, though. It brings up a discussion and makes you think about things. And, they are right. It's an engine of growth for the economy. That's one of the reasons why a number of years ago there was a huge increase to the NIH budget, right? Because it was going to fuel development and more jobs and things like that, more technology.
I mean, I agree. It should be even more.
Yeah.
I mean, we are throwing away money by not investing more in NIH because the return on investment is great.
Yep.
Right.
You know, every FDA-approved drug basically started with NIH money. So, and, you know, the CAR T therapy, I'm sure, started with NIH money. So,
Oh, yeah.
I don't know why they wouldn't invest more. Right? $35 billion they could easily put $100 billion into this.
Yep.
But they don't, and so you have to find other ways. All right, let's wrap up with some picks.
Sure.
What do you got, Steph?
So there's an app that you can, I haven't added it on my phone yet, but I saw it on Twitter and it looks very cool. You can hold your camera over plants and animals around you and it will ID them for you. I just think this is so cool. I mean, I'm constantly, hiking or walking and I have that thought, oh, what is that? I got to look that up. And so you can just point your phone at it and, and it's out of the California Academy of Sciences. So I don't know how far along this app is in IDing things. Let's say I'm in North Carolina or in New York or up in Ithaca, but it seems really interesting. So I'm going to try it.
In North Carolina, go ahead.
There's wildlife in North Carolina?
Yeah. Oh my gosh. So much wildlife.
There's poison ivy.
Yeah. Yes, there's it. There definitely is a lot of it in my backyard.
This is very cool. That's a great idea.
Yeah.
So that goes for plants and insects and wild pigs. Will it do a wild pig?
I don't know about maybe if I can get that close to a wild pig. I don't know if I could be able to tell that story.
That's pretty cool. I've heard of that somewhere. I don't remember where. Cindy, what do you have?
I have an interesting article that was published on Science website, and it's called Academia's Hard Work, but the taking a break is effort well spent too. And I like a couple of quotes from this article, and it says, taking a break is also hard work, and it's true because we think that we need to work really, really hard all the time, and the harder we work, the more successful we'll be. But when you think about it, when do your best ideas come? Both of you.
When I'm walking my dogs.
Bingo.
Yeah, doing other things. Yeah,
right.
When you have time to think about other things.
When your brain is off, right, and it's just kind of running in the background, humming along, and all of a sudden, bam, there the idea comes, right? But we spend so much time just slogging along and not giving our brain the chance to do that. It says the novel ideas don't come from a mind constantly under pressure. It says, my best ideas and aha moments almost always come after I allow my brain to relax and drift. And so the argument here is that, the harder you work, the more hours you spend in the lab, you fill in your notebooks and you get praised for how hard you're working and FaceTime that you're spending in the lab. But the reality is you need to balance that. You need to balance it. I'm not saying everybody should go on vacation every other week, but taking the time to just sit and think about your data, or even just don't think about your data. Things eventually come to you. I always joke, you wake up in the middle of the night, you're like, oh, I got it. You write it down, right? So it's just when you're trying not to think about it, that's when things come. And so I think it's not valued enough. One of the big things is sabbatical leaves, right? It's not a huge thing here, at least not in my department or in the College of Veterinary Medicine. Very few people do it, but it's something that why not? Why not? Why not take that, if you're given that opportunity, to take a few months and just sit and really, truly think. And so as an academic, it's more like when you were in graduate school, right? Like all you had to do really was think into experiments. And now we have all this other stuff, and it just gets more and more and more as you go along, and you get less and less time to actually think. And I keep saying to myself, if I only had time to sit and read those papers and really, really, really think about it, there's something there, and I just can't put my finger on it,
Well, that's why I podcast. I get to think about other things, and it gives me ideas.
Yeah.
It's highly recommended. You should try it, Cindy. Oh, you do?
Oh, you do?
I do.
Okay.
Yeah. So, Vincent, what do you have?
I have immunology posters free.
Ooh.
Oh, I love them.
From R&D Systems. B-cell development, inflammasomes, integrins, TGF-beta. Oh, a bunch of free posters with all kinds of pathways and interesting things. Cancer immunotherapy strategies, airway inflammation and asthma. So It's all from R&D Systems. Very cool.
TLRs are on there. Yay.
And you can have a PDF where you can download the PDF. It's big, and if you have a big printer, you could print it out or you could buy one. But anyway, it's cool.
Yeah, very cool.
I love these. I always pick them up at meetings and I put them up in the lab.
Yes.
When you have nothing to do, you're waiting, the centrifuge is spinning. If you put it behind the centrifuge, the people just kind of stare at it and they pick something up.
That's the time you should be
not
doing
thinking.
what you're still,
Yeah.
Take a break.
Watching the countdown. 4 minutes left, 3 minutes left.
All right. That is Immune number 19. You can find it on your phone and your tablet where you have a podcast player. And every phone has one, iOS or Android. And if you do listen, please subscribe so we know how many people are listening. It gives us a clue of who we're reaching. And as I said earlier, if you really love what we do, consider supporting us. Microbe.tv/contribute. You could give $1 a month, and that would really help if all of you did that. And questions and comments, go to immune@microbe.tv. Cindy Leifer can be found at Cornell University or on Twitter as Cindy Leifer. Thank you, Cindy.
Thank you.
Steph Langel's down south at Duke University. Not the deep south, but it's south.
No, it's south.
Stephanie Langel on Twitter. Thank you.
Yeah, thanks. This is fun.
I'm Vincent Racaniello. You can find me at virology.ws or Prof VRR on Twitter. Music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.